In today's video, we shall discuss about immunofluorescence. Before that, do subscribe to my YouTube channel and let me know your comments in the comment box. Immunofluorescence is a type of assay performed on biological samples to detect specific antigens in any biological specimen or sample and vice versa. The specificity of antibodies to their antigen is the base for immunofluorescence. It was described in 1942 and refined by Coons in 1950, which used a fluorescence microscope able to read the specific immunological reaction and cellular slide preparations. Principle of immunofluorescence. The property of certain dyes absorbing light rays at one particular wavelength and emitting them at a different wavelength is known as fluorescence. In the immunofluorescence test, a fluorescent dye that illuminates in UV light is used to detect or show the specific combination of an antigen and antibody. The dye usually used is fluorescein isothiocyanate, which gives yellow-green fluorescence. Specific antibodies bind to the protein or antigen of interest. Antibodies could be labeled with molecules that have the property of fluorescence. When light of one wavelength falls on fluorochrome, it absorbs that light to emit light of another wavelength. The emitted light can be viewed with a fluorescence microscope. Immunofluorescence tests are also termed fluorescent antibody tests. So why is immunofluorescence test used? The immunofluorescence is a histochemical laboratory staining technique that uses the specificity of antibodies to their antigen. It is widely used in immunohistochemistry based on the use of some fluorochromes to visualize the location of the antibodies. There are two types of immunofluorescence tests: direct immunofluorescence test and indirect immunofluorescence test. Direct immunofluorescence test. Direct immunofluorescence is a technique used to detect antigens or other biomolecules directly on a tissue or cell sample using a fluorescently labeled primary antibody. Direct if involves using a single labeled antibody that recognizes the target molecule, while indirect if involves using two antibodies. A primary antibody that recognizes the target molecule and a secondary antibody that is labeled with a fluorescent probe and recognizes the primary antibody. Procedure of direct immunofluorescence test. The mechanism of DF involves the following steps. Sample collection and preparation. The first step involves obtaining the tissue sample or cell specimen. The sample is then embedded in a suitable medium or embedded in paraffin. Sample sectioning. The embedded tissue is sliced into thin sections using a cryostat or a microtome. Fixation. The tissue sections are treated with a fixing agent, such as acetone or formaldehyde, to preserve the antigens and structural integrity of the tissue. This step is essential to prevent antigen degradation during the subsequent procedures. Blocking. To minimize non-specific binding of antibodies, the tissue sections are incubated with a blocking agent, typically bovine serum albumin or non-immune serum. Primary antibody incubation. A specific primary antibody is added to the tissue sections. This primary antibody recognizes and binds to the antigen of interest, if present in the tissue. The primary antibody is often conjugated with a fluorescent dye, such as FITC, fluorescein isothiocyanate, or rhodamine. Washing. After the primary antibody incubation, excess unbound antibodies are washed away from the tissue sections. This step helps to remove any unbound or non-specifically bound antibodies. Visualization. The tissue sections are examined under a fluorescence microscope equipped with appropriate filters to detect the emitted fluorescence from the labeled primary antibodies. If the target antigen is present in the tissue, it will fluoresce, indicating a positive result. Indirect immunofluorescence test. Indirect immunofluorescence is a technique used to detect and localize specific biomolecules, such as proteins or nucleic acids, within cells or tissues. IIF uses a primary antibody that specifically binds to the target antigen, followed by a secondary antibody that is labeled with a fluorescent probe. The primary antibody is not labeled and a fluorochrome-labeled secondary antibody is used for detection. The antigen used is known and it binds to the specific primary antibodies of interest in the sample. The secondary antibody then binds to the Fc region of the primary antibody. Procedure of indirect immunofluorescence test. The mechanism of IIF involves the following steps. Sample preparation. The sample is fixed onto a slide and treated to maintain the structural integrity of the cells or tissue. Primary antibody incubation. A primary antibody specific to the target antigen is added to the sample and allowed to bind to the antigen. Washing. The sample is washed to remove any unbound primary antibodies. Secondary antibody incubation. A secondary antibody that recognizes the primary antibody is added to the sample and allowed to bind to the primary antibody. Washing. The sample is washed again to remove any unbound secondary antibodies. Imaging. The sample is then visualized using a fluorescence microscope, which detects the fluorescence emitted by the labeled secondary antibody. The intensity and location of the fluorescence signal can be used to determine the location and quantity of the target antigen within the sample. This is a diagram of primary and secondary immunofluorescence. Primary immunofluorescence involves an antibody with a fluorophore emitting a signal once bound to a specific antigen's epitope. Secondary immunofluorescence has a secondary antibody with a fluorophore binding a primary antibody specific to the antigen's epitope. These are the image results of immunofluorescence tests. Applications of immunofluorescence. Diagnosis of autoimmune diseases. IIF is commonly used in the diagnosis of autoimmune diseases such as lupus, rheumatoid arthritis, and Sjogren's syndrome. Cancer diagnosis. IIF can be used to detect the presence of specific biomolecules, such as tumor markers or oncogenes, in cancer cells or tissues. Detection of infectious agents. IIF can be used to detect the presence of viral, bacterial, or fungal antigens in patient samples. Research. IIF is commonly used in research to investigate the expression and localization of specific biomolecules within cells or tissues. Limitations of immunofluorescence. The main problem can be photobleaching, which is the degradation of fluorochromes. It can be prevented by using higher concentration of fluorochromes and decreasing exposure time to the light. Extraneous unnecessary fluorescence can occur due to impurity of targeted antigen. Autofluorescence can occur due to some agents which bear the property of fluorescence in the given specimen. It is mostly used for only fixed cells or dead cells. Expensive and require higher expertise. To conclude, immunofluorescence is a powerful technique that allows researchers to visualize and localize specific molecules within cells and tissues using fluorescently labeled antibodies. This technique has revolutionized the field of cell biology by enabling the visualization of complex cellular structures and signaling pathways at the subcellular level. Immunofluorescence has numerous applications in biomedical research, including the diagnosis of diseases, the identification of cellular targets for drug development, and the study of cellular interactions in disease pathogenesis.